Hello students, in this video lecture I am going to explain you the process of semi-conservative replication of DNA. First of all, let us understand what you mean by the term replication of DNA. It can be defined as the process in which DNA molecules make exact copies of themselves. It is also called DNA duplication. Simply saying, it is a process by which one molecule of DNA produce two identical copies or replicas of itself. Please remember here that DNA is the only biological molecule which has got the unique property of self-replication, that is the ability to reproduce itself. Now, what is the significance of this process? Replication is essential to ensure the faithful transmission of the genetic material, that is the DNA, of the mother cell to daughter cells during cell division. Replication is again a enzymatic process. Many enzymes are needed to complete this process, but DNA polymerase is the key enzyme required. And also know that this process takes place during the S or the synthetic phase of the cell cycle. Please recollect, cell cycle divided into two phases, the mitotic phase and the resting or interphase. Resting phase is again divided into G1, S and G2. S synthetic phase is characterized by the process of DNA duplication or replication. Replication begins on the DNA molecule at several points which are called the replication origin. From this point, the DNA double strand unwinds or separates from each other and replication proceeds in both the directions. Therefore, we say replication is bidirectional. During replication, many Solon points called replication bubbles are formed along the length of a DNA molecule. And part of a replication bubble, which appears characteristically Y-shaped, is called a replication fork. This replication fork is the basic unit to study the process of DNA replication. Now let us try to understand all these concepts with the help of a diagram. You can see here, this is the DNA double molecule, and all along the length can you see these points which are called the origin of replication. Now the double stranded DNA has separated in these points, and this separation has resulted in the formation of bubble-like structures. So this is called as the replication bubble. So you can see many replication bubbles along the length of the DNA, and from this point of origin you can see these arrow marks directing on either direction. That means replication proceeds on both directions. So we say it is bidirectional. So here further progress has taken place, and ultimately you can see here two daughter DNA molecules are formed. So this is the structure of a replication fork. You can see here, this is the part of the DNA double helix which is not separated from each other, whereas this is the part of the DNA double helix where separation has taken place, and each of the separated strand has formed one new molecule. So in total it appears as a Y-shaped structure. So that is called as a replication fork, and this is the basic unit of replication which helps us in studying this process. Now another important aspect is that the DNA replication is called semi-conservative replication. So let us try to understand why it is called semi-conservative. Now this is a parent DNA molecule made up of two helically coiled strands. So now you can see here the DNA is producing its copies. Now this is the area where replication has started. Now the replication has completed and it has resulted in the formation of two daughter molecules. But to look at the color code here, the blue strand represents the DNA strand which has come from the parent DNA molecule, whereas the red coded strand represents the newly synthesized DNA molecule. Now please note here, each of the daughter DNA molecule has got one old strand, that is one blue strand, and one new strand, that is the red strand. So which means 50% of the old DNA is present in the new daughter DNA molecules, or 50% of the old DNA is conserved or retained in the new DNA molecules. So because of this, the replication is called a semi-conservative process. Now, as I said, replication requires several enzymes. Let us try to understand the functions of each of these enzymes. The first set of enzymes needed are called DNA helicase and DNA gyrase or topoisomerase. What they do is separation of the double strands. So the parent double helix unwinds by the action of these enzymes. And the next protein required is called single strand binding protein. Now they do a very specific job here. What they actually do is they bind to the separated strands of the parent DNA and prevent these separated strands recoiling. So therefore they are also called as single strand binding proteins. So to summarize, they actually stabilize the separated single strands. Now the next enzyme is called RNA primase enzyme. Now this primase enzyme synthesizes a short RNA molecule at the beginning of each new strand formation. Now this small RNA molecule is called as RNA primer, and this primer formation is a very important step in synthesis of a new strand. The most important enzyme required is called DNA polymerase. Of course, there are three types of DNA polymerases: polymerase 1, polymerase 2 and polymerase 3. Each of them have got specific functions to perform during replication. After that, DNA polymerase 3 is the most important enzyme, which is the enzyme actually responsible for the synthesis of a new DNA strand. So it synthesizes a new DNA strand by polymerization of complementary nucleotides complementary to that of the old strand, by adding nucleotide triphosphates to the 3' end of the new strand. That means a new DNA strand can form only in the 5', 3' direction, not in the reverse, that is 3', 5' direction. Please understand this. And finally, there is an enzyme which is called DNA ligase, which is required for joining small pieces of DNA molecules which are formed during DNA replication. Now, these small fragments are called as Okazaki fragments. And the last enzyme required is DNA polymerase 1, which actually removes the RNA primers which are formed at the beginning of each fragment, and these RNA nucleotides are then replaced by DNA nucleotides by DNA polymerase